Today I shall discuss on regulation of gene expression. So gene expression is a combined process of the transcription and the translation of any gene in the form of protein. So this is our DNA. What is happening here after being transcription of a particular gene, it is converted up into the messenger RNA. And this process is known as transcription. And after the transcription, messenger RNA comes into the cytoplasm here over the ribosome in association with the endoplasmic reticulum. It forms the protein. Desired kind of protein is being synthesized. So overall expression of the gene is in the form of protein. So the regulation of gene expression is essential for metabolic functions, for growth and development and differentiation of the tissue, because at every level we require any different kind of protein. So the direct link of gene expression is with that of protein. That manifestation is in the form of protein because proteins are responsible for various metabolic functions, for the growth and for development, for sustenance of life. We can say in the nutshell. So the rate of expression of prokaryotic gene is controlled mainly at the level of transcription. That is the messenger RNA synthesis. So when we will see the gene expression among the prokaryotes, so it's mainly regulated at this step, at the level of transcription. However, in the eukaryotes, there is a much more complex process of the genome control, and the gene expression in eukaryote is controlled by not one but many ways. So we will discuss later on. So the types of gene regulation, there are two types of gene regulation. One is the positive regulation, another one is a negative regulation. Positive means when the expression of genetic information is increased by presence of a specific regulatory element. So that regulation is known as positive. We will give certain example. You will better understand how it is a positive and how it is a negative regulation. So positive regulators are the molecules which regulate it in a positive manner. They are known as inducer. They are enhancing or they are uh, facilitating the expression of gene. So these are known as activator or inducer. While on the other hand, opposite to the positive, the negative regulation here in this expression of gene information is diminished by the presence of a specific regulatory element. And regulation is said to be negative. And the element mediating this negative regulation are called repressors. So keep remember these two names, activator or inducers and repressors. Both work in opposite manner. Now, types of genes, there are two types of gene. One is the inducible gene, another one is the constitutive gene. So what is the inducible gene? Inducible genes are expressed only when there is some inducer, some induction is there. So I will give you the example of production of enzymes beta galactoside by the presence of lactose in prokaryote. So I will tell you the concept of lac operon, how it is being operated. That will be the good example of inducible gene. Then second example is the insulin is an inducer of gene glucokinase of glycolysis in human beings. So these are two burning example. Then second is the constitutive gene. What is a constitutive gene? The constitutive gene refers to genes whose expression is not regulated. They don't require any regulation. They are expressed at constant rate all the time. They are never switched off. Only conditionally they are being inhibited for time being. So these are often referred to as housekeeping genes. Housekeeping genes. They keep a watch onto the functions of the cell. Then regulation of gene expression in prokaryotes. How it is being regulated. So in prokaryotes, the gene involved in the metabolic pathway are often present in a linear fashion, and that is known as operon. Operon is a unit, is a regulatory, is the what we call it. It is a regulatory It is a gene expression unit, and it does consist of regulatory gene, consist of control site, and consist of structural genes. So this whole unit is known as operon, what we call as operon. So operon is the unit, or the it is a genes involved in metabolic pathway are often present in this manner, and that processing unit is known as operon. So here I will give you the example of lac operon. Lac for lactose, and matlab the operon, or the this gene expression unit meant for lactose metabolism. So we will see the schematic diagram of the lac operon showing its protein. So this is this unit, this I. This is lac I, inhibitory zone, or like there is a these are the this is the regulatory gene. This is regulatory gene. Then second unit is control. It does consist of two regions. One is the promoter region, second is the operator region. Then these are the structural genes which are to be expressed once there is a command. So Z, Y, and A. And these and this whole gene unit is polycistronic, because in the E. coli, this whole unit is polycistronic, because on being expressed, the same, there are different zone of the gene responsible for synthesis of three different kind of proteins. So these three enzyme, beta galactosidase, permease, and trans-acetylase, they are expressed by this polycistronic LAC messenger, or this lac operon structural genes, right? So this operon is actually the lac operon, and is actually a group of genes. One side you have seen, these are the regulatory genes and these are the structural genes. So this is a group of genes with single promoter and single operator, and they are meant for transport and metabolism of lactose in E. coli and other bacteria. So these basically relish on glucose. But once the lactose is present, there is a need of its metabolism, its conversion to galactose and glucose. So, like in the presence of glucose and absence of lactose, so in such medium, this operon is in the switched off mode. We will see how it is in switched off mode, because already glucose is present in the medium, so there is no need of means, and there is like no presence of lactose, so the E. coli will keep on feeding on glucose. So how it is switched off, we will see it, because these are what we call, these genes are all time constant genes. So this is the repressor, the transcription of this gene, regulatory genes, that leads to the formation of repressor messenger RNA. And this repressor messenger RNA will synthesize a repressor protein that is in the active form, and it is now attached to the operon region. So once the operon region is locked, so the RNA polymerase, even if it is attached to the promoter region, it is no more able to reach to the actual structural gene site, and it will prevent or inhibit the expression of these genes. So repressor bind to the operator and prevents the transcription of Z, Y, and A genes. Right. So this is like a molecular switches. These repressor proteins, they are switching off the transcription of these structural genes, which are responsible for metabolization of lactose. Because lactose is not present, so it is to be inhibited. In the second case, the regulation of lac operon in the presence of lactose and absence of glucose. So once there is a absence of glucose, so it needs glucose. The bacteria needs glucose. Glucose will come from lactose. So lactose, there is a need of metabolism or digestion of lactose. So for this, what will happen? The reverse phenomenon will happen. Here the transcription is happening because it is all time happening. These are the constant gene. So repressor messenger RNA is being synthesized and leading to the formation of repressor protein. But this protein is now locked with lactose. Lactose as act as an inducer, and this is inducer repressor complex, and this make it inactive. It's no more able to attach onto the operon region. This operon region will remain open. So what will happen? This catabolite activator protein and cyclic AMP complex is now able to attach to this particular promoter region side. And this association will actually facilitate the winding of this RNA polymerase and its movements towards the structure gene site where there has to be where their transcription has to happen. So this cyclic AMP is also the concentration of cyclic AMP is also increased in the presence of lactose, which is being inhibited by glucose, right? So we can see it here now. RNA polymerase is ready to slide over to the actual sites of transcription. And now here is happening the transcription. Z, Y, and A gene are transcribing into the form of these protein beta galactosidase, then permease and trans-acetylase. Beta galactoside will splice or it will hydrolyze the lactose into the galactose and glucose. Permease will allow its entry into the cell, entry of the lactose into the cell, while trans-acetylase has a known function, but somehow it facilitates or supports the metabolization of this lactose. So this is about the regulation of this lack of operon in the presence and absence of lactose. Now, let's see the gene expression in the eukaryotes. It actually starts at the level of DNA and ends with an enzyme catalyzing a particular chemical reaction or with a structural or metabolic protein. So, gene expression in eukaryotes is controlled at 5 levels. One is at the level of DNA through replication, and second is transcriptional. At the level of transcription, there is a regulation. Then post-transcriptional at the level of synthesis, post-transcription of messenger RNA, and fourth at the level of translation, and fifth at the level of post-translational modification of the proteins. So we'll see one by one. Now, first the gene regulation at the level of DNA. So genes are altered by what are the processes by which the gene can be altered? It is through gene amplification or through gene rearrangement or the gene loss or through some a covalent modification of the DNA through like chemical modification of DNA through methylation, most common. So we will see here, this is an example of rearrangement of DNA. We know that immunoglobins are, a variety of immunoglobins, millions and billions kinds of immunoglobin the body can synthesize. They are quite unique and specific to a particular kind of pathogen. So how this, capability of the body to synthesize these huge variety of immunoglobulin it is because of only the process of rearrangement of DNA. So we can see it here. This is the gene region responsible for the expression of immunoglobulin. And we know immunoglobulin the protein has two regions. One is the constant region, another one is the variable region. Constant region has the constant type of protein. There is no change in the consistency of this protein. So constant region remains the same. Only it is the change in the variable region of this immunoglobulin, and that identifies that better fits to a pathogen. So this variable region has to be changed. How it is changed? It is changed at the level of altered gene expression of that particular region responsible for production of that variable region protein. So this is the region responsible for the expression of gene which lead to the synthesis of variable region protein. So there are various domains of this gene, and these are the variable domains and D1, D2, D3, etc. domains, and J1, J2, etc. And there are chances of rearrangement of these region depending upon the type of no, response received from the type of pathogen and how the variable region will better recognize and better fit to a pathogen to opsonize to that pathogen. So this is all through the variation. So there can be, millions of no types of combinations of these different domains or subsections of this gene, and that leads to the a unique kind of combination which is responsible for expression of heavy gene heavy chain gene of the variable region. So this is how every time a new kind of immunoglobulin with a variable region, a unique variable region and a constant region is being synthesized. So this is the rearrangement of DNA. Then second is the transcriptional regulation. So a major method of regulation in eukaryote is transcriptional regulation, which is manifested at the two levels in the eukaryotes. One is through chromosomal packaging and second is individual gene regulation. So we can see here in this particular diagram, so there are certain transcriptional factors. This is just an example. There can be many ways of regulations. You have just seen in the lack of operon also there was one way of transcriptional regulation, how the repressors are being arrested and how the repressors are activated. So now here in this eukaryotic transcriptional regulation, there are two zones, enhancers and promoter region, and the two transcriptional factors now they are attaching to this particular site and they lead to some conformational change in such a manner they pack the chromosome in such a way so that that association will facilitate the attachment of RNA polymerase. So this is how the transcriptional regulation is maintained through chromosomal packaging. Then at the level of post-transcriptional regulation, so these mechanisms control or regulate the messenger RNA after it has been produced. So post-transcriptional processing of RNA is much more extensive in eukaryotes than in prokaryotes. So there are two ways. One is the alternative splicing and regulation of RNA stability. Then next is at the level of translational regulation at the level of translation. So most eukaryotic translational control affect the initiation of protein synthesis and the initiation factors required for translation, particularly the elongation factors too, involved in these regulatory mechanism. So action of these elongation factors is inhibited when it is phosphorylated by some protein kinases. So this is one way of how it is being regulated. Then post-translational control, this mechanism act after the protein has been produced. So these were the various ways with few examples of regulation of gene expression in the eukaryotes. Thank you.